Welcome guys to the first Flip Classroom lecture of the year. Today we're going to be talking about the
scientific method.
And everything that you see us writing, you guys need to also write on your Cornell notes because it's going to help you with your second project of the year, which is going to be due at the end of the week. So let's dive in. What do we need to know about the scientific method?
Well, let's start with the fact that there is no one scientific method. You guys have probably learned a certain number of steps in the past that your teachers have told you is the scientific method. And what we're telling you now is that there is no one scientific method. There are a bunch of components that you'll find in every good experiment and every good article, but you might not find them in exactly the order or the particular context that you've seen them in before. So let's start with what are some of the components. Well, the first one is kind of self-explanatory, and that is some kind of problem.
Yeah, so typically when you're reading a journal or an article, it's going to start out by saying, well, this is what we see. Or this is what we're trying to figure out. Why is this going on? So in order for us to even know that a problem exists in the world, we need to make some
observations.
And so observations and problems are used interchangeably in science.
Right.
So what is the definition of an observation?
So observations are basically using your senses to gather data. Okay. Some kind of data.
Okay.
And there are two different kinds of data that we want you guys to really know about.
And the two types are qualitative
and quantitative.
All right, so we're going to go ahead and write down the definition so that you guys can write it down. And when we talk about your senses, we're talking about the 5 senses that people generally think of. So the ability to see, hear, smell, taste, and touch. And so those 5 senses are going to help us collect 2 types of observations or 2 types of data, and those 2 types of data are, again, qualitative data and quantitative data. And we're going to start out with qualitative first. So let's talk about what qualitative data is.
So one of the best ways to remember these 2 words is to kind of look at their stems. And this word has the stem qualit or quality in it. And so qualitative data basically is using your senses to gather information that is a quality or a description. So if we were looking at the room behind us, we might say that the room was blue. We might say that the drapery was white. We might say that the pillow over there is soft to touch. So all of those things are descriptions or qualities of what we are looking at.
So in your research that you guys are going to be looking at, and we're going to be looking at lots of different research this year, we want you to be able to tell us the difference between qualitative data that's presented and also quantitative data. And we want you guys to be aware of what we're looking for when we ask you to do an experiment. So we might be only interested in qualitative data in one experiment and only quantitative data in another. And if you give us the wrong type of data, obviously you're going to lose a significant amount of points, and we don't want that to happen. So let's talk about quantitative data.
So quantitative data, really think the best thing to do is focus on that N, because that N indicates that we are looking for something that starts with
an N.
Numbers! Right, so we're looking for data that has a numerical context. So we might say that this room has 2 windows, or that we have 10 fingers, or if we're looking at chimps, that they are 5.2 meters high.
Okay, so now that we know the difference between the types of data that need to be collected or the types of observations that can be done, we can say or address a problem even exists. So now that we have a problem that exists, the next step is to
come up with some kind of hypothesis. And hypothesis is a word that I'm sure you've heard many times in science, and so we're going to go ahead and define it right now. So what is the definition of a hypothesis?
Generally, we refer to as a hypothesis as an educated guess.
What does that mean, educated guess?
So that means in order to make an educated guess, you have to have made some observations. So a hypothesis relies on good qualitative and quantitative observations. You have to have a sense of what's going on around you in order to make an assumption or an inference about what you think might happen.
Oh, you just said a word I don't know if I know. Inference. What is an inference?
So an inference is basically using prior knowledge and observations that you've collected to make an assumption or a guess about what you think might be happening.
Ah, so the word inference and hypothesis are sometimes used interchangeably?
That's exactly right.
Ah.
And so in your scientific literature, and that's the point here, we want you to be scientifically literate to be able to understand what's going on in scientific journals. If you see inference or if you see hypothesis, they're talking about the same thing.
Okay. So now that I have my hypothesis or my inference, I need to take it a step further because I'm going to want to eventually kind of test this to figure out what's going on with my problem. So I might make what's called a prediction. And predictions are pretty important because they're basically taking your hypothesis and turning that into a statement about what you think will happen based on your guess.
Yeah, so it's kind of a cause and effect statement. Now, in order for that cause and effect statement to be perfect in the scientific world, we need it to be set up in a very specific way. So there's a formula for writing a prediction.
Right. So we always start our predictions out with if. So if this one thing happens, and we'll come back to tell you what you put into that spot later, but if this thing happens, then this other thing is going to happen as a result. So see, we have that cause and effect. So if is the cause, and then is the effect.
Okay, so we've dealt with a problem. We have to make observations to figure out whether or not a problem exists, and then we have our two types of observations, qualitative and quantitative. The next thing is a hypothesis, also known as
an inference,
which is just an educated guess. And then typically what would happen is a scientist would then set up a cause and effect situation, but it's written in a very specific way.
Right, the if and the then statement.
So the next thing that we want to talk about is?
The next thing we want to talk about is the controlled experiment. In other words, what we are doing to test our prediction or our hypothesis.
Now, there's a reason why we call it a controlled experiment and not just
an experiment.
So how can an experiment truly be controlled? Because as researchers, we want to study very specific things, so we don't want other things to enter into our experiment that we can't control for or account for. So in a good experiment, there are always a certain number of groups.
Right. We have two groups. We have what's called the experimental group and we have what's called the control group. And both of them are really important. And we want you to be able to find and pick those out of a particular article. And some experiments may have even more than one experimental group, but you will always have just one control group.
So let's start out with the experimental group first. So we know that we're always going to have one control group, but some experiments have hundreds of different groups.
Right.
And so that means if there were 100 groups and we know one of them is the control group, all of the rest of those groups are then what?
The experimental group.
So let's talk about what differentiates those two groups. Is it easy to understand the difference between the two? Can you spot it easily in research?
Well, I think so. Because really the experiment is what you are testing, or is the test. So the experimental group is the group that is receiving the test. Or, in other words, our independent variable, but we'll get to that in a second. But the experimental group is the group that is being tested on.
Okay, so it's the group that's receiving the experiment.
Right.
Okay, so the experimental group, if you want to find it in your reading, and you're going to have to do this today, and really for the rest of the year, the one thing that you need to be thinking about, if you're looking for the experimental group, you're looking for the group that's having something done to it.
Right. So they are getting the treatment or being tested upon.
And I think that Creasy mentioned earlier that treatment or that test is referred to as the IV. So let's go ahead and put it in our definition, and we're going to spend some time talking about IVs and DVs here in a few minutes. But now that we know what an experimental group is, let's talk about a control.
So a control group is really important, and it's just the opposite. It is the group that is not receiving the test. So really they are not being tested on. You are not giving them your independent variable, and that's really important because that is allowing you to collect a baseline or normal data for that particular group. Why would it be really important to collect a normal or baseline?
Well, if we just had experimental groups in an experiment, we wouldn't be able to tell whether or not there were any differences, or what the deviation is, how far from normal those experimental groups actually traveled because of the manipulation or the event or the research that we were trying to do. So our control group is the normal group. It's the group that we don't do anything to. That's the key. Whenever you're looking for the control, find a group that has not been changed. It's just doing what it always does. And the reason why we need it is because we need it to compare.
That's a key word there. Comparison. So the control group is what's allowing you to compare it versus the experimental group to see if there has been a change. Because if there has been a change, and if you've set your experiment up correctly, that change should have something to do with your independent variable.
And so we have a simple chant so that you don't forget it. This is what we always tell people. When you're reading an experiment and you're trying to figure out what the experimental group is versus the control group is easy to find because it's the thing that stays the same, you need it to compare.
Right.
The thing that stays the same, you need it to compare. So whenever we ask you, and we're going to ask you today,
Oh, yes.
tell us what the experimental groups are, tell us what the control group is. Once you find the control group, that means everything else is the what?
Experimental group.
So it's really easy to wrap that question up because you're just looking for the group that hasn't received anything unusual.
Right.
All right, so now that we've talked about the two types of groups, let's talk about this weird thing, this IV versus the DV.
Right. So variable. So variables are things that vary or things that change. And so your independent variable, which starts with an I, is very easy to remember because it is the variable that I change or that the researcher changes.
Yeah, so as Creasy's writing this down on the screen so you guys can put this in your notes, the independent variable typically when you're reading an experiment, put yourself in the experiment. So if the experiment is saying the researchers did X, Y, and Z, cross out the researchers and put your own name in there. Ask yourself, if I was running the experiment, what would I be changing? What am I manipulating? Sometimes they use that word instead of the word change in the experiment. So they might say in the abstract, which is the very beginning, this is what we manipulated in the experiment because.
Right.
And then you could read on to answer some other types of questions. So now let's talk about the dependent variable.
So the dependent variable is depending upon the independent variable. That's where it gets that name dependent. And it is really easy to pick out because it's usually a variable that is being measured by you or the researcher. So you're looking for something that is in units, some kind of units usually. So it's the thing that is being measured.
So the dependent variable is basically the data.
Yes.
That's another way of thinking about it too. So the independent variable is what I'm changing, and then what I change impacts what I'm going to measure.
Right. Of course it would. And that is, again, why we have that term dependent. It is depending or changing because of the independent variable.
Okay, so let's just briefly wrap this up to make sure everybody understands what a controlled experiment is. How many minimum groups should we have?
So minimum you should have two. You should have the experimental and you should have the control group. And when you are trying to find them, again, the experimental group, think experiment, they're the group that's being tested on, receiving the treatment.
Mm-hmm. And then why do we even need a control group then?
Normal. You're getting the baseline data or the normal data so that you can compare the control group that doesn't have anything done to them with the experimental group, what you tested on, to see if there's been a difference. So comparison is that key word there.
Okay, and then we have our IV versus our DV. The IV is the?
The IV is what is changing. What is changing because I am changing it, or you, or the researcher. So the thing that I change is the independent variable.
And then that leads us with the very last thing, which is the dependent variable.
And that, again, is what you're measuring. So that's depending upon the independent variable and changing because of it, and therefore being measured in some kind of units. So it's the data.
Okay, so now that we've talked about this third part, the controlled experiment, that leads us to the very last thing that we're going to need you to understand, because you're going to see it, and typically comes at the end of a research paper.
Right.
Typically.
Right, and that's the conclusion. And the conclusion is really important because the conclusion basically explains a lot of things. Why the experiment was important, what data was gathered during that experiment, whether that data was key to the hypothesis, does it support the hypothesis, does it not support the hypothesis, and really explains what the findings of the experiment were.
Okay, so let's go back and focus on the most important things that Creasy just said. So the conclusion is going to be the findings. What did we figure out? What was the whole point of the experiment? But more importantly, the conclusion also connects the data or the controlled experiment back up to the hypothesis.
Right, exactly.
So our hypothesis was our educated guess, what we thought might happen if we link it to a prediction. So in order to figure out whether or not our hypothesis should be rejected or accepted, we need to see whether or not the data or our measurements support what we thought was going to happen.
Right. So when we look at that data, we really want to look at it in relation to the hypothesis. So connect your data with the hypothesis. Say whether or not it supported it or rejected it.
And if you notice that the data doesn't support your hypothesis, which, to be honest with you, happens more times than not, that means that you have to go back and you have to redo the experiment.
Right.
And that's okay because science is this messy, messy, messy thing. And so oftentimes what you'll see is that A researcher will spend years trying to figure out the answer to one question and run potentially hundreds or even thousands of experiments because every time they've run it, it's not supported the hypothesis.
Well, and also, keep in mind that every single experiment should really only have one variable that is changing. So it might not be that variable that causes the desired outcome. So you might have to run it again and again and again with all these different changes to figure out which one is doing what you want it to do. And you can't do them all at once because that would be really confusing and it wouldn't give you that good experimental design and control. So really, that's often why we see experiments lasting lots of time, because one variable is very important to having a good experiment.
So I think that we might need to add just one little addendum at the bottom of our screen here that Creasy just brought up, which is really important. When we talked about IVs and DVs, we never really mentioned, well, how many of them do you have? And we know that a DV, you can have tons of them, right? Because it's just what your findings, the information that you're collecting. But in a really good experiment, we should only have one IV.
Right.
Because if we had anything more than just one thing that we're changing, if we actually saw a change, we wouldn't be able to figure out, well, what caused it, because we changed more than one thing. So a good experiment only has one independent variable, and I guarantee you that's one of the key indicators to determine whether or not the experiment you're reading about is any good.
Yeah, definitely.
And when you guys set up an experiment today, wink, wink, because you're going to have to do it, we're looking for one IV. If you're giving us anything more than one, you and your group have made a major mistake, and you're going to have to go back and start again.
Right.
Okay, so now that we've given you all the information that you need to be successful in your second project of the year, please make sure that you filled in everything. If you have any questions, let us know. And then throughout the course of completing your project,
you can go back. You can watch this movie as many times as you need to. It's on the internet for a reason. We want you to have it in your toolbox so that it helps you get where you need to get with your experiment.
Okay, thanks guys. Good luck with your projects.
Good luck.